Welcome to the International Whaling Commission's Cetacean Strandings Investigation training videos. This video introduces personal safety, preparation and sample storage during a cetacean strandings investigation. Cetaceans can carry zoonotic diseases that pose risks to humans. Before beginning any animal examination, it is essential to have appropriate personal protection equipment, or PPE, to protect against exposure or injury. If PPE is not available, no examination should be undertaken, and the animal should instead be just photographed. Photographs are a vital investigation resource and should also be taken at every stage in any examination. PPE should provide coverage appropriate for the local climate. You should have waterproof overalls and footwear to protect against fluids, grease and blood. Waterproof overalls must be over waterproof boots and not tucked in. Proper footwear also reduces the risk of slipping on wet or greasy surfaces. Gloves are essential and must be worn at all times when touching the carcass or handling samples. It is recommended to wear arm-length gloves with cut-resistant gloves on each hand for added protection, and surgical gloves layered over them. Eye cover is also advisable. Avoid touching your face or personal items to prevent cross-contamination. Before starting, prepare your necropsy toolkit. Make sure that all bags and sample pots are labelled in advance and made to include the unique identifying number on every item. Ensure each sample is labelled with a unique identifier for the case, the tissue type, and where applicable, the storage medium. Seek advice from your local team on the correct unique identifier. If starting from scratch, choose a code short enough to be written on all samples that include species and date. Where possible, best practice items include spare gloves, cutting board, sharp knives, ideally with a guard to prevent hand slippage onto the blade, a knife sharpener, scissors, forceps, tape measure, ruler, and a camera with a spare memory card and battery. You will also need bags and pots to store samples, food-grade foil for wrapping samples, and where applicable, storage mediums such as formalin or ethanol. Store tools securely when not in use and transport them in a designated toolbox. Always ensure tools are clean and keep blades sharp to ensure clean cuts and minimize the risk of slips. Before beginning, agree on roles. At least one person should remain clean to handle pots, samples, and notes. PPE must still be worn. Be clear about which samples you'll collect and how they will be stored. Tissue can be taken to a clean surface to make cutting down to container size easier. Always handle tissue carefully to avoid introducing crush artifacts. It is important to remember that every sample is labelled with three things: the unique case identifier, the tissue type, and where applicable, the storage medium. Tissue samples that are not stored in formalin should be chilled or frozen as soon as possible to slow decomposition and preserve diagnostic information. It is important to keep samples as cold as possible between the necropsy site and the storage facility. In the next video, we will cover the steps for conducting a basic external examination of the animal.